So I will start now. Today I will be talking about patterns of development. So patterns which are present, developmental patterns essentially, specifications, and all these things we are going to be studying today. For that, let me take an attendance. Control Shift S. I have to send all these to the principal. Okay, I am sharing my screen. So mechanism of developmental patterning, that is specifying identity. This is a bit of this particular PDF. It's a mashup from Gilbert and some of my notes and other things. So I've underlined it, highlighted regions which you need to study, and you people must concentrate on those regions itself. Nothing else on these regions which have been underlined. The rest you can study, but I don't think you people will be willing to do so. Okay. So one of the America's first embryologists, William Keith Brooks, he reflected that the greatest of all wonders of the material universe, the existence in a simple, unorganized egg of a power to produce a definite adult animal. So it is really a strangely wonderful event that in this universe, from one egg, a small egg, which is unorganized, that is, nothing is regular inside this egg, it has got a power to form a particular definite individual. For in our case, say for example, we have got one head, two arms, two legs, and two eyes, two ears, and that is what you call as organized. So what will be this chapter will introduce the concept of cell specification. That is from unorganized to organized. They become from unorganized to organized. That is, a particular cell becomes specified to form either a nerve cell, it can form a muscle cell. So, and this is known as, it is specified to form a type of cell. So this chapter will study about all these things. So let us talk about the first thing, that is cell differentiation. So what is cell differentiation? The generation of specialized cell types is called differentiation. And it is a process during which a cell ceases to divide and develop specialized structural elements and distinct functional properties. That is, say for example, a cell is differentiating to become a nerve cell. And differentiation, it is the process during which you see the nerve. The first, what happens, the neuroblastoma cell, it keeps on the neuroglial nerve precursor cell, it keeps on dividing. Now it wants to differentiate. What it does before differentiation, it actually stops dividing, and then certain specialized structures like axons and all, they come out, and they have got a distinct functional property. That is, they carry different voltage along their they carry the voltage along the axons, and they carry electrical impulse. So it has got a distinct functional property. Previously, what happened? It started from one cell. That cell could have become anything, but then it became differentiated. Process of differentiation. By the process of differentiation, first what it happened? It stopped dividing, and then it developed specialized structural elements, and then distinct functional properties. So three things which you see in differentiation. First হচ্ছে division stops. The second is that develops specialized structural elements. Specialized structural elements. And third is it's got distinct functional properties. So these are the three things which happen. For example, consider a red blood cell. It differs in its protein composition and cell structure from an eye lens cell or a neuron in the brain. It's different. The proteins are different. But before differentiation, there is a step known as commitment. Before differentiation comes the step of commitment. That is, the cell commits to a certain fate. The cell may not look different from its neighboring cell, but it is committed. That means that cell, although this nerve precursor cell is same as looking as the other cell, but this nerve precursor cell is differentiated to form nerve cell only. It is committed. So before differentiates and looks different, it may look the same, but already inside several processes have started, and that is known as commitment. It's like almost like, for example, you have a boyfriend or a girlfriend. So you are committed. That means you are not yet married. Differentiation is the marriage. But when you walk on the road, you look like a girl who is not committed, or a boy who is not committed, who doesn't have girlfriend or a boyfriend. But they are not committed. But they look the same. But already inside that person who has got a girlfriend or boyfriend, commitment has taken place. That is that this person will not get interested by any other boy or girl, committed to one person. So same, this cell, it may look like the neighboring cells, but it already has it is going in the process towards forming a nerve or a muscle or any other organ. So this is the difference between the pre-step of commitment. So this commitment has also got two stages. The first is specification. That is, the fate of a cell or tissue is said to be specified when it is capable of differentiating autonomously, that is by itself, when placed in an environment that is neutral with respect to the developmental pathway, such as a petri dish. So let us look at a figure. Say these are committed cells. Basically, they have falsely added a brown color and a yellow color, but they are looking the same. They are, because the color which is given is falsely given, just to differentiate. So the cell is isolated. If it's this cell is isolated and put in a petri dish here, and the other cell is isolated and put over here. This cell is already specified to be a muscle, and this cell is specified to a neuron. So in a petri dish, provided you give it food and all stuff like that, this is going to be differentiated into a muscle cell here, and this into a nerve cell over here. This is what you mean by it is specification. First step, specification. That is capable of differentiating autonomously. Differentiating autonomously. Capable of differentiating autonomously. At this stage of the specification, inside commitment also, they have just met for 2 days, or they have met for 10 days. If they have met for 2 days, the girl and the boy, then maybe their relationship is not that strong. So it can be altered, provided they are given a different, another handsome or more beautiful girl or a much more better girl comes, they exchange partners again. They break up and new partners are formed. This stage is known as specification. That is, if a specified cell is transplanted to a population of differently specified cells, the fate of the transplant will be altered by its interaction with its new neighbors. This can be seen in this figure. Now, this muscle cell is placed in between nerve cells. What happens? It was already specified for making a nerve cell, but it gets changed into a nerve cell. Instead of muscle, it forms a nerve cell inside. When this specification is reversible, the specification is labile before. But when this specification is irreversible, this cell will be a muscle cell wherever you place it. That is known as determined. Determination. The cell is already determined. Cell is already determined to become a muscle despite the surrounding environment. And this determination, this is the second stage of commitment. So first stage is specification. Second step is determination. Commitment can be divided into specification and determination. In specification, if you place them independently, if there are better partners don't come, they will marry eventually. But when they are determined in specifications, but they will marry. That is in specification. But suddenly you find put them in a batch of very nice boys, they will not marry. They will get determined with somebody else. So this is how you can define the difference between specification and determination. In determination, however good boys you put around, it is going to the muscle cell which is specified for committed for making muscle cell only, the muscle cell precursor which is committed for making muscle cell only, it will become a muscle cell. Now this type of specification, the first step, it can change. Oh, before that, maybe I'll explain a term known as trochoblast. Trochophore is an echinoderm mollusk. There are trochophore larvae, molluscan larva, trochophore. You must have read it in your invertebrates, trochophore larva. So these trochophore larvae have got cells which have got cilia on the surface. They have cilia. They are trochophore cells. And the cells which will give rise to this cilia-borne cells, they are known as trochoblast cells. I'll actually give the examples right now. So once, before I explain something, during specification, there are three major strategies that embryos can exhibit. One is autonomous, conditional, and syncytial. So now this is clear that first specification takes place, and then determination takes place, and then finally the shape changes and it becomes differentiation. So three steps. Specification. Then the next step is determination. They are determined. And finally the marriage or differentiation. So these are the strategies of cell commitment. One major strategy is autonomous specification. One type of specification. Strategy of specification. The blastomeres of the early embryo they have got a set of critical determination factors within the egg cytoplasm. So blastomeres, you know, this is the first, the sperm and the egg fuse and they form a zygote. They divide and form a ball, and each cell is known as a blastomere. Each cell is a blastomere. So what happens is that each cell, right from the zygote, how is the zygote formed? You have the egg and the sperm comes and fertilizes and it forms the zygote over here. So this zygote, inside this zygote, it is already provided with a set of determination factors. Determination factors, and they are known as morphogenetic determinants. So inside the zygote, say this is a morphogenetic determinant over here. Let's take a different color. Another morphogenetic determinant is over here. Let's take another color. Another morphogenetic determinant is over here in this, in the egg, in the zygote. This is the zygote. It's present like that. What happens is, eventually, say for example, this region, and this region, they will become different cells. They will become different cells. This will be a different cell. This will be a different cell, and this will be a different cell. This is at one type of cell, second type of cell, and this is a third type of cell. So they will be forming different types of cells. Example I will give you of a snail known as patella. It is a snail, a mollusk, phylum Mollusca. Now, in the normal development of patella, what happens? This is the cell. These are the presumptive procoblasts over here. This flesh colored, this is one presumptive trochoblast. This is the second presumptive trochoblast. This is the third presumptive trochoblast. Already the trochoblast has not yet formed. So they keep on, the cells keep on dividing here, keeping on dividing here. All the cells are dividing. And finally, you find these ciliated trochoblast cells formed in this trochophore larva. Due to the formation of trochophorula, these ciliated cells are formed. Already the cells over in this region, first what it was a single cell. Already in this region, and this region, they had some morphogenetic determinants which told that these cells will form trochoblast. Already something was present inside the cell. Then the cell divided already in this region. Let's use a lighter color here. Already in this region, this region and this region, there was a morphogenetic determinant already present, which told that this will form trophoblast cells with cilia. Now, for example, if you isolate this cell, it will divide and form the ciliated cells. Ciliated cells are formed. This happens on its own, by itself. Therefore, it is known as autonomous specification. So, already this region is autonomously specified to form ciliated cells. Next thing which I will do, this is the part where I had played God on top of Gilbert's book. I've taken something from another famous book known as Slack, another famous book, Slack. So, what is a cytoplasmic determinant? So, a cytoplasmic determinant is a substance located in a part of an egg or blastomere that guarantees the assumption of a particular state of commitment by the cell which inherited it during cleavage. So it is the same as morphogenetic determinant, but it is present exclusively in the cytoplasm. So it is the same as morphogenetic determinants. A cytoplasmic determinant, because it is present in a part of an egg or a blastomere, and it guarantees that this particular part is going to become a head or a tail or a muscle or whatever. So this is the operation of a cytoplasmic determinant coding for anterior structure. Operation of a cytoplasmic determinant coding for the anterior structure of the embryo. In normal development, it ensures that the head forms from the cells that inherit it. So, if you transplant the determinant, that is this region, is supposed to form the head. It's transplanted to another region. You will see in that organism an ectopic head or two heads are formed. Here it is not transplanted, the normal organism is formed. This region will form the head. It's over there, a second head is formed. That is because of the presence of certain cytoplasmic determinants which are present in this region. If you pluck it from here and put it over here, two heads will be formed. This is cytoplasmic determinant. Some factor, normally they are transcription factors. Normally they are transcription factors. I have cut out all these places. You don't have to study these. Now we go to the next part, which is conditional specification. So conditional specification, it is the process by which cells achieve their respective fates by interacting with each other. That is, if one cell is over here and the surrounding cells say so, all these cells, they interact with each other. They involve cell-to-cell contacts. And the mechanism is known as juxtacrine. So factors flow from one cell to the other cell and they influence each other or they specify each other. Like these light blue cells, they may specify the green to become another blue cell. Can very well happen. The famous experiment which I just told right now. This is the blastula. In this region are the normal back cells. Okay, so cells from black back tissue, no transplantation. You see a normal development. Then this region is transplanted into this region. This region will form in this region. If cells are removed from the embryo, the remaining cells can regulate and compensate for the missing part. Say, you remove these cells. The surrounding cells, they have got certain factors. They will compensate the missing part. So conditional specification is the process by which cells achieve their respective fates by interacting with the other. Juxtacrine factor, secreted signals. So if this, for example, this region, this red, this region is removed. Surrounding cells, they compensate that region and they form that cells. Because one or two cells, they have got these conditional determinants or morphogenetic determinants. They will work hard and they will make this thing perfect. So conditional specification is what a cell becomes depends on its position in the embryo. Its fate is determined by interactions with the neighboring cells. So, if the neighboring cells are nerve cells in blue, I'm drawing them in blue, then there is a high possibility that this green cell will become blue. Where it is. If it's between nerve cells, it will become a nerve cell. If it's between muscle cells, it will become muscle cells. Or if it's between cells which have got muscle cell determinants, it's going to become muscle cell determinants. Rox had made an attempt to demonstrate autonomous specification. In the two-cell stage, he destroyed one cell, dead tissue, living tissue. Only half the embryo was formed. If you remove it, then it will become twins. But if you kill one side, only half the embryo will be formed. This is autonomous specification. Because this side was specified to form the other half. It's killed. If it's killed, that side is still attached but killed. Only half has got embryo will form. This side will not form anything. But for example, if you remove this area, then the living area that will compensate and make the other area. This is what we learned from here, normal development. Next, we go to syncytial specification. It is both autonomous and conditional. Autonomous is whatever the surrounding be, it is all present inside. मेरे अंदर है, मुझे ये करना है. And conditional is मेरे अंदर कुछ नहीं है, बाहर से जो बोल रहे हैं मैं वही कर रहा हूं. You people are under in a phase of autonomous specification. However hard I'm telling you that you people, your careers are doomed until or unless you pay attention to the careers council services. You people are autonomously determined to become sweepers on the railway platform, because it is inside you that I have to become a railway platform sweeper. However hard I'm trying to tell you or make you understand that I'm giving so many conditions, you are not listening. You're ignoring, because you people are very clever. So this cleverness is your autonomous specification. And conditional specification, that is something that, say for example, my former students in batches before your batch, they were conditional specified. They listened to whatever I had to say from my experience and what to do it. They did it. So today, one of your seniors, Sanjay Das, is about to go to not very early 2019 pass out. He is going to Germany or Switzerland possibly to do his PhD. He's risen to this level. So this is conditional specification. Apart from this, there is a strategy which uses both. This is known as syncytial specification. Possibly you people could have done both of this, taken both these ideas together and then done something. That would have been ideal. Your own ideas inside to do, but your ideas is to become a railway sweeper, railway platform sweeper. So you're welcome to be whatever. All professions are noble. So a notable example of an embryo that goes through a syncytial stage is found in insects. It is found in Drosophila. So let us see the development of Drosophila. This is the pre-blastoderm cycles. 1 to 9. Stages 1 to 9. The red dots are nuclei which are dividing. They are calling it purple over here, but I am looking at it as red. So the red dots are nuclei. They are dividing. What is a syncytium? The nuclei divides, but the cytoplasm doesn't divide. There is no barrier between the nuclei. So in one cell, you have many nuclei forming. Cycles 4 to 6, it's like this. Cycle 8 to 9, the nuclei migrate towards cortex. Cortex means the surrounding border. This is the border is the cortex. Outside region is cortex, inner region is medulla. From kidneys, you may know. Then cycle 8 to 9. The nucleus migrates towards the cortex, and then cycle 10 to 13. A syncytium is that there is a division of the nucleus, but the cytoplasm doesn't divide. So in one cell, you'll find many nucleus. Insect development shows these stages. This is the early development of. Then the next thing is, after this is then cellularization takes place. It takes place after this syncytial blastoderm is formed. Then cellularization takes place, and then the cellular blastoderm is formed. Unlike other cells, non-insects, where one zygote divides to form 2 cell, then 4 cell states, 8 cell states, 16 cell states, 32 cell states, 64 cell states. Then forms a blastula, morula, gastrulation. It's not like that. In insects, it's the nucleus keeps on dividing inside. This is the egg, fertilized egg. Inside this egg, this starts to take place. Syncytium. Inside this egg, as you know, there are that famous cytoplasmic determinants. So let us look what happens. Have a look at this picture. Let me increase the size. You see two colors. And you see two names. One is bicoid and the other is caudal. One is bicoid. So the red color is caudal. Sorry, the reddish color is bicoid and the bluish color is caudal. The anterior side of the egg, and this is the posterior side of the egg. Already the egg is there. You have a higher concentration. This is a gradient, high concentration to low concentration. This is a gradient. More towards the anterior side, you have higher concentration of bicoid, and gradually in this higher concentration of caudal, and it's like a gradient. इस साइड में ज्यादा है, इस साइड में ब्लू कम है, इस साइड में रेड ज्यादा है, इस साइड में रेड कम है. A gradient exists already inside the egg before. Then the nuclei keeps on dividing to form a syncytium. And this nuclear genes, they are activated by morphogen gradients or cytoplasmic gradients or cytoplasmic determinants. All these words are the same: morphological gradient, morphological determinant. They are calling it as a gradient. These determinants are called as a gradient because their concentration is not uniform. Towards one end it is high, and as it goes towards the center, their concentration becomes lighter and lighter and lighter till it disappears on this side. And inside this side, so what does these are transcription factors or determinants. Join করলো. So these are determinants. So genes, nuclei in this region, they are exposed much more to bicoid, and they become specified for the making cells of the anterior end, like head and all these things. And nuclei in this region, or the genes in the nuclei in this region, they are more exposed to caudal. Here they are less exposed in the center. So depending upon the gradient concentration of bicoid or caudal, depending upon the bicoid and caudal, they are becoming specified into posterior and anterior end. Cellularization takes place, and this region forms the head, this region forms the thorax, this region forms the abdomen, or then this region forms the tail. So Be sure to read about this very nicely. Let's So, do you people have any questions? Please do ask your questions if you have. কোনো প্রশ্ন থাকলে জিজ্ঞেস করো। Any questions? শুনতে পাচ্ছ?
হ্যাঁ স্যার। হ্যাঁ স্যার।
কোনো প্রশ্ন আছে? ওকে,
এটা পড়ে নি।
না স্যার, আপাতত তো স্যার নেই, এটা একবার পড়ে নিয়ে
হ্যাঁ পড়ে নাও, আমি পাঠিয়ে দিয়েছি হোয়াটসঅ্যাপে।
হ্যাঁ স্যার দেখলাম। হ্যাঁ
পড়ে নাও, পড়ে নিয়ে প্রশ্ন থাকলে জিজ্ঞেস করো।
স্যার নেক্সট ক্লাস স্যার কবে নেবেন?
আমি তোমাদের বলে দেবো, আমার অনেক কলেজের কাজে তো ব্যস্ত থাকি।